Welcome to this installment of the Classics of Immunology Journal Club. And on YouTube, if you like this video, go ahead and subscribe. Click on the little bell to get notified. And each time we upload a new video, you will be notified. And then all of the links are below for my website, and you can find all the classic papers of immunology there. Happy reading. So today we're going to go into a paper that was published in 1967 by a fellow by the name of Donald Mosier. We've had a string of papers that are single author papers, which is not the usual thing today. But Don happens to be a contemporary of mine. Give you a little background about him. He was a student when I was a student, and he happened to be an MD PhD student at the University of Chicago, and he was working with a pathologist by the name of Donald Rowley, who was an old-time really good professor at the University of Chicago. And I'm sure that whoever had the bright idea about this paper and so forth, they probably had both a lot to do for it. But it was nice that Rowley allowed this young whippersnapper to become a single author on a paper in Science magazine in 1967. And the title of this paper is a requirement for two cell types for antibody formation in vitro. And so last week we did the Michel and Dutton method of generating what they thought was a primary immune response in vitro by mixing together unimmunized mouse spleen cells or lymph node cells with sheep red blood cells as an antigen, and culturing them together for a few days, and then assaying the resultant cells to see whether or not they were making antibody. And in order to do that, they used the plaque assay that had been reported a few years previously by Niels Jerne, the Jerne plaque assay. So this paper is an example, basically a very prime example of why it was so groundbreaking and path-breaking for these in vitro culture methods to be used in immunology, because for the first time they could start to take apart all the different parts of all the cells and so forth, and then ultimately the molecules that were really participating in what we thought and what we called an immune response. In the very first sentence of this paper is only 2 or 3 pages long, so this is another one you can go read about. And the very first sentence goes as the following. It says, antibody synthesis may result from the interaction of two functionally different cell types, one which phagocytoses and processes the antigen to provide the stimulus for the second type, the lymphoid cells, which synthesize the specific antibody. Now that sentence is what the paper says. So I don't know why they let off with that. They reference Don references several papers to support the idea that this might be a reasonable hypothesis based on what the observations were that are out there. If you read these papers, there's 3 or 4 of them. It was a lot of cigar waving, really, essentially, that maybe there were 2 different kinds of cells involved in this whole business of synthesizing antibodies, but there was no clear data. And one of the major problems is that it was because the experiments that were available up until this point in time were mostly in vivo, and it was difficult, as I've said before, to take apart the Swiss clock and put all the parts out onto the table and then find out what time it was, because the Swiss clock didn't work anymore. So this whole business of in vitro cellular immunology was the thing that broke the field wide open. And so what they say in the introduction is that they decided to use the Michel-Dutton system to look into this whole situation. And so most of the paper is really the materials and methods that they used, as well as the results that they got out of this whole thing. And then with a little bit of discussion and conjecture at the very end of the paper. So they used the Michel-Dutton system almost exactly the same as Michel and Dutton had reported it. And of course, they reported it in Science in 1966, and then followed that with a much longer methods paper that we dealt with last week in 1967. So they used splenocytes from unimmunized mice. They had them at a very high density of 10 to the 7th, 10 million cells per milliliter. They used 35 millimeter Petri dishes that we talked about last week. They used a low oxygen tension of 7%. They fed these cultures daily for 1, 2, 3, 4, 5 days, and with a cocktail mixture of essential, non-essential amino acids, and supplemented with special batches of fetal calf serum. Wouldn't work without that. And they started with the hypothesis that the 2 different cells involved that they thought in this whole process. One were plastic or glass adherents so that they could use that to separate the cells one from another. The lymphoid cells or the lymphocytes were not adherent to glass, and you could stick them on into a flask or a petri dish for eons, and they would not attach. By comparison, the macrophages would attach and then spread out onto the glass like fried eggs, and you couldn't get them off. To get them off, you had to scrape them off with what they called a rubber policeman, which is just a glass rod with a little rubber tip on the end. You scrape them off the dish. You lost a few doing that, but for the most part, you could get them off of there doing that whole thing. So what they did in order to separate the lymphoid cells from the adherent cells, which they called the A cells, was they simply take the spleen cells and dissociate them into a single cell suspension, put them onto the plastic or the glass, and wait for 30 minutes in the incubator, 37 degrees. And then they'd come back and then would very gently pipette off the cells that were non-adherent. And then you could take them and put them onto another petri dish or flask and just repeat the process. And every time you do that, you get more and more and more cells to stick to the plastic, and the lymphoid cells would be passed up into the pipette and so forth. And so they then would place these cells into culture, either separately, the adherent cells versus the lymphoid cells. And when they did that, and then waited 2, 3, 4, 5 days, and then did the urinary plaque assay at the end, they got no antibody formation. So the conclusion was, you've got to have both of these cells, maybe. That was then shown when they put them back together again, then they would get antibody formation in vitro. So that was good. That was sort of supported their hypothesis that both of these cells were really essential to this whole process. And then they did experiments whereby they took the adherent cells and also the lymphoid cells, and incubated them with the antigen, the sheep red blood cells, independently, and for 20, 30 minutes, 37 degrees in the incubator. And they looked at it under the microscope, and they found that the adherent cells would take up the red blood cells. You could see them inside the adherent cells, and it looked like only about 5% of the macrophages within that time period, 30-minute time period, would take up sheep red blood cells. But the interesting thing was, then that would work. That's all you needed, was that 5% inside the sheep red blood cells. That then led to the interpretation that there was something going on with the macrophages and the adherent cells and the antigen that was ultimately became called antigen processing. And so that then worked. When they did that, they could still get the same number of plaque forming cells, antibody forming cells out of the whole thing. So that was it. That, you didn't have to do, back in the day, to get into science or nature is pretty easy. You just do a few experiments and you're a student, you throw it in there, and you become famous overnight. So that was it. That was the end of the paper. And the thing is that it really, it begs the question, or it begged the question at that point in time. So the conclusion was, we need two different kinds of cells to make antibodies. We need lymphocytes and we need macrophages. Now the lymphocytes, that would become the next question. Because don't forget, we've already seen Jacques Miller's experiments in the early 60s, 1961 and on, where he said that there was something about the thymus that was really important. They knew that the thymus, and therefore the cells derived from the thymus, didn't make the antibodies. But subsequently, what's happened in this business is that there wasn't just 2 cells that were required for this. It was really 3 cells. You needed the T cells for the B cells to make antibody. Now, me being a T cell person, I always said, well, obviously, yeah, the B cells don't know what to do. They have to be told. They have to give by the T cells because they know what's going on, essentially. So we'll get into that. But the very next year, in 1968, there was a paper from Joe Oppenheim, who's an old friend of mine, and he was a young guy then too. And he basically showed that in order to get a proliferative response from lymphoid cells, from the peripheral blood or from the spleen and the lymph node, that you needed macrophages for that. Now, that was really before B cells and T cells had been recognized and defined, but that's coming up soon in our discussions of the classics of immunology. Things started to move very fast now after 1965. In 1965, we realized we had the first lymphokines, and then we had interferon, and now we're getting more cells involved in the whole antibody forming response. And so things really started to hop in the late 60s. And so I'll stop there. Don't forget, if you really like this video, subscribe, get yourself notified by clicking on the bell, and check out all the links below, because you'll find them how to get all these papers for the Classics Journal Club, as well as other things on my website. So thank you. It's been great.